about is microbial in the environment. Microbes are omnipresent in the biosphere. Omnipresent meaning they are present everywhere. And their presence is actually invariably affects the environment. The effects of microbes on their environment can be beneficial or harmful or inapparent with regard to human measure or observation. The most significant effect of microbes on Earth is their ability to recycle the primary elements that make up all living systems, especially the carbon, nitrogen, and oxygen. Primary production, which involves photosynthetic organisms such as they take carbon dioxide from the atmosphere and convert it to organic material. The process is also called carbon fixation, and it accounts for a very large portion of organic carbon available for synthesis of cell material. Decomposition of biodegradation results in the breakdown of complex organic materials to other forms of carbon that can be used to other organisms. Environmental communities and their factors. The group of similar species create population which result in a community. Microbial community means all microbial population in a habitat. The activities of complex communities of microbes affect biogeochemical transformation in natural, managed and engineered ecosystem. Microbial communities are very important for the rigorous progress in the field of agriculture, which increase the rate of crop production. Microbial community may be terrestrial or aquatic. In terrestrial communities, a community of microbes and their environment that occurs on the land masses of continents and islands form a terrestrial microenvironment. Terrestrial microenvironment is distinguished from the aquatic microenvironment ecosystem by the lower availability of water and the consequent importance of water as a limiting factor. Rainforests are the most diverse and productive terrestrial microenvironment, but their soil is nutrient deficiency due to extensive leaching by rainwater. Soil formation is a slow process that involves tissue, chemical withering, and biological processes. Over millions of years, microbes play an important role in soil. They aggregate formation and soil stability that confer fertility and productivity to soil. The soil microbes participate in this process through many ways. Filamentous microbes assemble clay particles using extensive network of hyphae resulting into soil aggregates. Additionally, some microbes secrete exopolysaccharide or cause compaction of clay particles that promote soil aggregation. The surface soil is always rich in indigenous population of bacteria, including actinomycetes, fungi, algae, and protozoa. The atmosphere is an inhospitable climate for microbes because of stress due to dehydration. This results in a limited time frame for microbes to be active. However, some microbes get resistance of these stresses through specific mechanisms, promoting loss of their biological activity. Spore-forming bacteria, molds, fungi, and cyst-forming protozoa all have specific mechanisms through which they are protected protected from this harsh gaseous environment. The relative humidity or relative water content of the air is critical for survival of the airborne microbes. Most of the gram-negative bacteria associated with aerosol are able to survive for longer period at low relative humidity, whereas in contrast, gram-positive bacteria remain viable longer in association with high relative humidity. The ability of microbes to survive in aerosol is related to the organism surface biochemistry. One possible explanation of this fact could be a structural change in lipid bilayers of the cell membrane in response to very low humidity. During loss of water, the cell membrane bilayer changes from the typical crystalline structure to a gel phase and affect the surface protein configuration, resulting into inactivation of the cell. Temperature is a critical factor influencing the activity of microbes. In general, high temperature leads to inactivation due to desiccation and protein denaturation, whereas lower temperature promotes longer survival rates. At the very low temperature, some microbes lose viability because of ice crystal formation on their surface due to freezing. Mostly, radiation at low wavelengths or UV radiation and ionic radiation or X-rays is harmful for microbes, causing DNA damage. This radiation target DNA by producing single or double strand break and changing the structure of nucleic acid bases. UV radiation causes damage by forming intrastrand thymidine dimers, causing inhibition of biological activity such as replication of genome, transcription, and translation. In aquatic communities, aquatic microenvironments occupy more than 70% of the Earth's surface, including mostly ocean, but also others such as estuaries, harbors, rivers, lake, wetlands, streams, springs, aquifers. The microbiota living in aquatic environment are the primary producer, responsible for approximately half of primary production on Earth, and primary consumers as well. A large variety of microbial community lives in aquatic environments such as the planktonic, sediment, microbial mat, biofilm community, etc. Planktons refers to photoautotrophic microbial community, including eukaryotic algae and prokaryotes, cyanobacteria, and heterotrophic community including bacteria or bacterioplankton, and protozoa or zooplankton. Phytoplanktons are the primary producer in the food web, using their ability to fix carbon dioxide into organic matter through photosynthesis. Aquatic microenvironment is further classified into three microenvironments occupied by microbes living in freshwater, brackish water, and marine water. There are two types of freshwater environment: standing water or lentic habitats such as lake, ponds, bogs, and running water or lotic habitats including springs, river, and streams. Lentic habitats are dominated by phytoplankton, forming distinct community gradient based upon the wavelengths and the amount of light that penetrates to a deep. Example is chlorobium. Chlorobium can utilize longer wavelength than other phototrophs and survive with little or no oxygen by consuming hydrogen sulfur instead of hydrogen oxide for photosynthesis. Marine water environments are highly diverse and contain 33 to 37% salinity. The ocean is divided into two zones on the basis of light ability: photic zone, where light can penetrate, and apoptic zone with lower light. In extremophilic communities, the organisms living in physical or geochemical extreme conditions that are detrimental to most life on Earth are termed as extremophiles. Most of the extremophiles are microbes and belong to the domain Archaea. Here are some extreme environmental conditions where extremophiles survive. Environment with high temperature, 70 degrees Celsius, including terrestrial and submarine spring with temperature of 100 degrees Celsius, hydrothermal vents with temperature more than 300 degrees Celsius are inhospitable for most forms of life, except some bacteria and archaea. For example, thermos, Methanobacterium, Sulfolobus, Pyrodictum, and Pyrococcus. Some organisms requires substantially higher than that found in seawater for their growth, and they are known as halotolerant. Halobacterium and haloanaerobium are two examples of halotolerant bacteria. However, some algae and fungi also exhibit halotolerance feature.